So now we're headed to another Power Pack talk next with a scientist, innovator, and bioengineer. Dr. Barry Lutz is associate professor in the Department of Bioengineering at UW. He's a Pilcher faculty fellow, and his expertise is in microfluidic physics applied to point of care diagnostics. And Dr. Lutz and his team developed the Rapid Harmony COVID-19 test. So thank you so much, Dr. Lutz. We are honored to have you here today, and over to you.
All right, can you hear me?
Yes.
And see screen?
Mhm.
Excellent. All right, well, thank you so much for having me here. I tell you, I've been so inspired by working with y'all so far, the fact that you have put this conference on and that so many young people are attending and interested in global health. So today I'm going to be talking about diagnostic tests for global health and maybe more specifically bioengineering innovation for what we call point of care diagnostics. So I've got a bunch of folks listed here, who've done a lot of work in the lab, but I wanted to call out Dr. Natada Pampradis in part because you'll see her name over and over again in today's presentation. But then also she's on a panel tomorrow, so you'll get to hear from her directly. So to start off, because many of you may be thinking about what you want to do. Obviously, you're exploring global health. I want to tell you a little bit about my pathway that wasn't really conventional. Unlike some other people who really knew what they wanted to do, I went to college unsure of what I wanted to study. When it came time to choose a major, it came down to just the fact that I had heard a cool story about chemical engineers who had developed some oil-eating bacteria. I thought it sounded cool, so I picked chemical engineering, almost on a whim. I didn't do well in school. I nearly failed out the first semester, but then slowly figured out how to do better and how to learn through the course of college. By the time it came to graduate school, I applied, just got into one PhD program, and that's because my undergraduate advisor advocated for me to get into the program. Glad that he did, because obviously that led me to where I am today. So my background PhD is in microfluidics. When I graduated there, I still wasn't quite sure what I wanted to do. Started to get into biology a little bit more, single cell microfluidics. Worked for Intel for a while to do cancer detection in tissue samples. And then Paul Yeager invited me to join his group. And he's University of Washington, very well-known professor in diagnostics for global health. So now, faculty in bioengineering, we continue to develop point of care diagnostics for global health, including collaborations with Paul Yeager. So I wouldn't recommend this type of approach for people in choosing a career path, but I do want to give people comfort that if you're still trying to feel that out, that keep trying, keep exploring, find what you want to do, and I'm sure that you will find your path. And I know just by being here today, you're already doing that, so that's very encouraging. Okay, so for today, in terms of topics, we'll start talk about diagnostics, point of care diagnostics. Why do we need these? What are they? And then in particular, how can we as bioengineers work with clinicians and other stakeholders to develop point of care diagnostics for global health? And this involves really an interdisciplinary approach, but even within the technology space, there's lots of different technical understanding that needs to go into bioengineering to do this type of work. I'll talk briefly about what it takes to, once you do something in lab, to actually get it out into real use. And then looking forward a little bit, what are the next things in terms of diagnostics that are on the horizon? So starting with a big picture of health problems, this is disability adjusted life years. This is basically health loss. So it involves death, but then also disability rolled into a single figure. If we look at high income countries, the blue conditions over here will be things that you recognize. So IHD is ischemic heart disease, stroke, we see diabetes, we see some mental health issues, Alzheimer's. So things that we're very familiar with as big important problems in high income countries. The red over here is mostly communicable diseases. So lower respiratory infections is the only one that really shows up there with a large figure. So it's a very relatively small problem for health in high income countries. So now if we look at middle income countries, this landscape changes. And you see that basically this red area got bigger. And as we go down to low income countries, you can see that it becomes very large. So the proportion of lost health in low income countries is disproportionately due to mostly these communicable diseases or infectious diseases. Fortunately, if you look over at the right, you'll see that some of these are coming down. I'll come back to that towards the end. But if we look at the global map and look at the health losses for communicable diseases, you can see that there are certain places, certain regions of the world where the contribution is significant. And here in the United States, of course, it's not something like tuberculosis we just don't think very much about. So to put this in perspective a little bit, tuberculosis is one of the biggest problems. Obviously, we've just been through this pandemic. Everybody, paid a lot of attention. We turned our lives upside down to deal with this pandemic. And tuberculosis kills people at a higher rate every year than COVID-19 has done over the past few years of the pandemic. So it is a pandemic, tuberculosis, and it's occurring every year in low-income countries, and yet we allow it to proceed. It's not an issue if we have good funding, if we have good healthcare systems, and it's a major issue where those resources are lacking. So overall, just big picture, main point here is that communicable diseases remain a significant problem in low-income countries, even though we feel like we have tackled them for the most part in the United States. So point of care diagnostics, what are they and why are they needed? So if we think of somebody who starts to exhibit symptoms in a low resource area, might visit a clinic. This might involve traveling for some time to get to the clinic. Hopefully they can see somebody with medical training. They might be asked to give a sample for sending off to a diagnostic laboratory. At that point, it's going to be a while. The sample has got to be sent off for testing. And it could be days or potentially even weeks before a test result comes back. And only when you get a test result can you make a decision about what type of treatment might be needed. So, think for a second about this scenario. You've gone into a clinic, the sample has been sent off to a laboratory. What kind of problems could this create to have this delay in getting a diagnosis and some intervention for the disease? So these could be effects on, the person who's sick, could be effects on the person's family, could be effects on the community. So a few of the things that are pretty key are obviously the person who is sick could get worse. Their condition could continue to progress. So the person's health is at risk. We know, of course, from COVID and other infectious diseases that we could have spread to other people. They could spread it to their family or their community members. So if we haven't intervened to isolate that person, to change their behavior, then disease progression or disease transmission could occur. There could be losses that are related to the fact that the person is sick and is not getting better as fast as they could. Lost work time, maybe they can't take their children to school. So there are societal losses, costs associated with people being ill that can be a factor as well. And then in some places, this is true, in some low income areas and in some communities in the United States as well, that you may lose contact with the patient. So they leave, and by the time the test result comes back, you're not able to locate the person to actually intervene and do something about it. So these are all significant costs that contribute to worse health and worse overall performance of society. So people obviously have been working on this problem in many different ways. I do want to emphasize I'm a bioengineer, so I tend to want to do technology solutions, but there are lots of different ways to try to address this problem. But from a technology perspective, a lot of work has been done. So I'm just going to show a quick video here about one approach. And actually, Mare, did you just hear some sound when I push the button? Are you able to hear that sound?
Yes, we were able to.
Great. Okay, I was hoping it would transmit. So this is just a little news clip about some technology that was developed to help transport samples from clinics to laboratories.
team in South Africa has been test flying these tiny uncrewed planes, which have been modified to carry blood and spit samples from remote areas to distant labs. They can be launched from clinics and follow pre-programmed routes using GPS and gyroscopes to orient themselves. Once they reach their destination, they can either automatically drop their parcel or respond to instructions from the ground. So far, 2 of these planes have been put to the test. Both of them successfully followed their routes and delivered the goods, even when confronted with wind speeds of up to 45 kilometers per hour. The larger of the 2 planes should be able to carry up to 500 grams, enough to transport many samples.
So that's a really interesting technology approach to try to bridge the gap between remote clinics and the laboratory. But what we have been interested in, and a lot of other people also, is point-of-care diagnostics. And this is really bringing the laboratory to the location of the patient. So the goal here is that you've got the doctor, the person with medical expertise, the patient, a test result, and the ability to give an intervention of some type or do an intervention all very quickly in one setting. And here an intervention could be a medication, it could be isolation to prevent transmission of disease, it could be that they need some surgical procedure, but the purpose of a diagnostic test is to give us information, give the healthcare provider information that they can actually act on to improve the health of the patient. Typically, diagnosing somebody, recognizing what the cause of their condition is, doesn't have high value if we can't do anything about it, right? So we want to tie this to an intervention. So in point of care, so a diagnostic could obviously be a laboratory test, but point of care diagnostic is generally a rapid result on the spot so that you can take immediate action. And that's important for, in terms of distinction for if there are diseases that progress slowly, so like cancer for example, you might not need to have a result today. It might be okay to send that off to a laboratory. But for infectious diseases, typically you want to intervene as quickly as possible. So how can we detect infectious diseases? There have been lots of methods developed over hundreds of years to detect organisms that cause disease. One of the classic methods is for bacteria or viruses that you try to grow them. You put them in an environment where they'll grow well, like in a petri dish, and you see if you get colonies forming. And you can use this potentially to identify what that organism is. So this really was the gold standard in terms of sensitivity for many different pathogens for a long time. Another way is to detect some protein that is unique to that pathogen. So the COVID test, the point of care test, the rapid test that you get are detecting a protein that is unique to the SARS-CoV-2 pathogen. So it has a unique shape that can be detected in those strip tests that you get. So these are broadly described as immunoassay tests. They use an antibody to recognize that protein. But really, what has been becoming the gold standard for many years is detection of the actual pathogen nucleic acid sequence or the genetic code, either the DNA or the RNA of that pathogen. And so a lot of you, I didn't learn PCR when I was in high school or learn about PCR, didn't hear about that until late into college. But some of y'all may have heard about it. This is polymerase chain reaction. It is basically an amplification method to sensitively detect RNA or DNA. And so this is where our work has been focused, and really it's where the gold standard is going, certainly in high income areas, but that high performance testing should be available. We want to make it available in low resource areas as well. So just as a quick little primer here, oh, no pun intended, this will just give you the basics of PCR. So basically we have a sequence that is from the organism that we're trying to detect. Here we're showing double-stranded DNA. So it means there are 2 strands of DNA. This is the A's, G's, C's, and T's that make up the genetic code. We would start the test in a laboratory setting by heating up the sample. Then there are things called primers that are small sequences of DNA that we add in that bind specifically to that target. There's a polymerase that creates new DNA. It extends that DNA to create a copy of it. And then that copies back the opposite direction. So every time we're creating two copies of the DNA, every time we heat up and cool down. We do this about 40 times typically, and that can go from having just about 100 copies or a few hundred copies up to billions or trillions of pieces of DNA that are copies of that original sequence. And then that's very easy for us to detect specifically to identify which organism is present. So that's the basics of PCR. That's what's done in the lab. When you do a laboratory test for COVID-19, it's this PCR process, again, as the gold standard for diagnostics. So in a lab, you can do this by having people who have good training and can prepare all of the chemicals that are needed to do this test, handle the samples carefully, process those samples, and then you have to have laboratory space and equipment to be able to carry out the PCR. You can also automate this, develop robotics or instruments that will automatically carry out all of these steps. And obviously there's a cost associated with that, both to make them and then also to keep them working. These types of things break. And so when we talk about point of care diagnostics for low resource settings, what we're trying to do is to think about how we can engineer these systems to make this high performance test type available and accessible in low resource settings. So that's where bioengineers and technology developers can potentially contribute by developing technologies that might meet these needs. So this is a lot of words, but some points that I want to get across here. When we're talking about these settings, they're really extraordinary constraints, especially for doing high performance or sophisticated biological measurements like we're talking about here. In some places, the electricity may be unreliable, so you may need battery power or no reliance on power. Refrigeration or freezing of chemicals. These tests use sensitive chemicals that normally in the laboratory we store in the freezer and the refrigerator, and then we prepare them when we're ready to use them. There may be less training than in a high resource laboratory, regardless of whether it's located here or anywhere else. Lack of equipment or disrepair of equipment could be an issue. Uncontrolled environment, the temperature control, dust control, things like that may be less than you would expect in a very polished laboratory. So to develop technology solutions, you really need to understand the engineering side of things, but then of course the health problem also. You need, what are the inputs to that problem that you're trying to address? And then the innovations that are done in the engineering space need to be designed to address these really extreme constraints. And so I think it's important, and I was glad that we heard this a couple of times this morning from Dr. Farquhar and Dr. Masayoko, that engaging local expertise and local stakeholders is obviously critical, to understand the problem, to get engagement, acceptance, and partnership, in developing technologies. So certainly that's the first part of the problem. The other side, or the complementary factor, is the technical expertise, and that's the part that I'm going to focus on in the next couple of slides. The sort of the basics of engineering, physics, chemistry, hardware development, electrical circuit development, software, those types of things that can contribute as components to a diagnostic device. Okay, so I'm just going to hit a couple of things. These are just kind of cool things to illustrate the technology side of it. Then I'll talk a little bit about some systems that have actually, the whole test systems that we have developed. You'll see that a lot of these things don't actually appear in those. And I'll mention why that might be here in a little bit. So this is a cool thing that we developed with Paul Jaeger. This is the idea that if you're trying to make a little machine that carries out steps of moving liquids around, how might you do that? And so this is a device that has a couple of strips of paper, and so that's these vertical strips right here that can suck up liquids, and we designate that these different different spaces are intended to carry out different chemical operations. And we want to bring fluid into one place, hold it there for a little bit, and then switch it or turn on a valve to move it to another place. So one of the neat tricks that we used in some cases was to use a dry sponge and activate that with water coming in. And that causes the sponge to expand, and that creates mechanical force that can then be used to push two pieces of paper together to create a contact and allow fluid to move from one space to the next. And then we could have another sponge that is activated at a different time to create the second step. And so that's what's shown over here at the right. So this is a wick, a different type of paper that sucks liquid up. It takes a certain amount of time for it to get to this first spot. And that activates the sponge and creates the connection here. That liquid then goes into a second timer over here and flows through and activates another sponge that then causes this last step to happen. So there are lots of different ways you could imagine. Maybe your imagination is running already with different ways that you could cause this type of timed actuation. Another approach just based on very simple wicking of fluid, just the phenomena that happens like when you get a paper towel and dip it into a puddle of liquid. You see the liquid getting pulled into the paper towel. This is exactly the same effect. We have a bunch of liquid tubes. These are just glass tubes that are filled with colored liquids that represent different chemicals that we're trying to flow across a reaction zone in a timed sequence. And so if we just touch this down to the dry piece of paper, you'll see that the liquid over here is the first to leave. The others just wait until the liquid in front of them goes, and then they start to drain away. So without any sort of extra mechanical motion or valving or switching, where this simple phenomena allows sequencing of these fluids in a timed sequence. So there are lots of other ways, I'm not going to show here, but to do this type of fluidic manipulation. On the chemistry side, I'll show three examples here. One is in the area of tuberculosis detection. And this is actually a project that Natada, Dr. Pampradis, came up with when she was a student at UW. For tuberculosis infection, we normally need to get a sample from deep in the lungs. It requires somebody to cough up a really nasty, thick, mucus-like sample. And then that has to go through pretty intense processing before it goes to PCR. And so the premise here that Natada and Dr. Morangu had was to look in urine for small fragments of DNA that come from the dying TB organisms. Those pieces of DNA can find their way into the bloodstream. Some of those pieces of DNA can go into the urine. And then you have, if you collect urine, you can try to find these small pieces of DNA. And so the technology approach that we took was to use a magnetic bead that had a strand of DNA connected to it that was complementary to the sequence of TB DNA that we were looking for. You mix these together, you get binding together of the target DNA with your capture, your bait DNA here. A magnet can be used to pull those out of solution. And then you can put those into a PCR reaction to amplify the target DNA. And so we actually have done this on clinical samples and shown that you can diagnose about 85% of TB patients from a urine sample. Second chemistry example is actually also from Natada's main project for single base mutation detection. So if we have a sequence here that has where the wild type indicates what we consider the normal pathogen sequence, and then there's a mutant sequence, so this could be COVID, in Natada's case, it has been HIV for the most part, for her project. So if we have a base that is changed here, we can have pieces of DNA that can stick to those sequences, and if they stick exactly, so they match, so the A is complementary to the T, that forms an actual pair that's intended to happen. Then we can stitch those two pieces of DNA together using an enzyme that now makes those two pieces of DNA into now one long piece of DNA. Whereas if there isn't a match, so the C base does not match, does not pair with T, then we will get no ligation. And so this is a transduction mechanism that allows us to ask, is there a mutation at this particular spot in the sequence that we're looking for? And for the case of HIV, this is important because this type of mutation, just one base change, can cause that HIV virus to be resistant to certain medications. So this test allows us to identify those mutations directly. Method is used this for other purposes, COVID-19 variant identification to figure out is it Omicron, is it Delta. Also for genotyping fish to understand fisheries and how if people are harvesting fish properly from certain waters. And you'll hear, I think, a little bit more about this on Thursday, or about her work on Thursday. Okay, third and last chemistry example is we're interested in, we always use protein. So I mentioned ligase as a protein. For PCR, there's a polymerase that's the enzyme that extends the DNA. And this molecule here that we see is an example of a polymerase. It was actually one that was created in our lab. So the goal of this work was to, first of all, create a new polymerase, but then try to change this polymerase so that it could work under harsh conditions. So it could work like if it got too hot, or it could work if it was in like a nasty environment like blood, or even a harsh chemical environment. And so the approach that we're taking, this is Ian Hull, a PhD student in the group, is to take this protein sequence, start to make mutations at a bunch of different locations in the protein, and then test to see whether it is able to function under these harsh conditions. So basically change it and see if it works. And so if this can be successful, it would allow a lot more freedom, flexibility, robustness to the PCR type assays in terms of the types of samples that they could work with and the types of conditions that they could withstand. Okay, on hardware side, we do a little bit of hardware development. And so this is just one example. This was also developed for the HIV project that I talked about. So this is a device that basically it has a small heater in it. So you can put tubes into this little aluminum block, and it will hold them at the right temperature, or potentially change temperatures if we want that to happen. It has light sources, filters, and light detectors in it so that we can hit the sample with a certain color of light. It will generate a fluorescence if the target sequence that we're looking for is present, and then that light gets filtered. And if that color light is detected, then we know that that target is present. And so this device here was built. It's pretty low cost device, but actually works quite well for detecting two different colors of fluorescence in the same sample. Then on the software side, the software can be used to provide instructions. It could be used for analyzing data so that the user doesn't have to do interpretation. This is an example, again, from Natida of a collaboration that she set up with Eric Clavins, who's the chair of electrical engineering in our department, to use software that they had developed for doing guided sophisticated laboratory procedures, and adapted it to guide people through the steps of the HIV drug resistance test. So you can see an example screenshot here of very simple instructions that it tracks the sample numbers as you go through, so the sample identifiers as you go through the process, and prompts every single step that you need to execute, because it can be very easy to make a mistake in a pipetting procedure. And then at the end, you get these strips that are kind of like the COVID rapid test that you get, except here, each one of the lines that we're looking at, the strips that we're looking at, is actually reporting whether that single base mutation is present or not. And so having a software to be able to interpret this and identify which drug the organism is susceptible to is very useful and works pretty well. Okay, so that's sort of just a rough survey of kind of the technical kind of examples of parts that can go into these systems. But sort of tying it together, this is the HIV drug resistance test. I think you know this by now, but it has PCR. This is done in a low-cost thermal cycler. It's essentially taking a really big, long, difficult, technically complex experiment, like a molecular biology procedure, and turning it into a simple kit with pretty simple instructions. So everything that's in the kit is shown on the left. These are reagents that don't have to, most of them, they don't have to be stored frozen. Some of them are refrigerated, but most of them can be stored at room temperature, which reduces the storage requirements and shipping requirements. It does this single base mutation detection with visual readouts, software guidance, automated interpretation, and then the results from this can actually be comparable to really sophisticated methods that you may know about called Sanger sequencing. So to have a very simple kit and simple procedure that can compete and give results comparable to a high performance laboratory sequencing method is a step forward. Second example is the test that Mayor mentioned. This is the COVID-19 test that we developed at the early stages of the pandemic. And again, here on the left is a test kit where the components are stored. These can be stored all at room temperature. They're in foil pouches to protect them from moisture. There's a sampling device down at the bottom left there. But everything that you would need to run a test is in that test kit. And then over at the right is a device. It's that heater fluorescence device that I showed earlier, connected to a cell phone, so that you can interface, you can control, you can look at the data, you can get the results, and you can input sample IDs. So I'll just show a video of how this operation works. So this is the cell phone interface. There's an app. It's written by Bob Atkinson to prompt you for all the steps. That would be the swabbing the nose, a step right there. This is all of the reagents that are needed for essentially PCR. We're doing a little bit different chemical reaction, but it's equivalent to PCR. That's cracking open a little barrier for liquid, a buffer to go into the sample. Now virus is coming off of the sample tip, and the dropper allows dropping a couple of drops of sample into that. It dissolves those reagents. And at that point, that's all you have to do. You don't have to go mix a bunch of chemicals or anything else. That's ready to go in the device. So here, they're scanning the ID on the sample so that it can know who is who going into that test. Tube goes in there to that little heater. You've got fancy LEDs. And now it begins the fluorescence measurement, much like what would happen in a fancy PCR system. So those are just a couple of examples of sort of complete systems that actually do allow you to conduct the entire process in a somewhat simplified manner. But I want to spend just a couple of minutes talking about this. A lot of times in academic work, we see these types of demonstrations, or maybe even less fully developed demonstrations that don't have all the pieces together. But even this type of system that has all the parts, it's really just the beginning of the process. Hopefully, and I didn't put this here, but hopefully before we started, we've engaged stakeholders and understood really what the problems and what the needs are so that the technology is correct. So that's an essential early stage thing to do. But once we get to this stage and we want to get this out so that it's going to be used, there are a couple of things that have to happen there. One is that somebody has to make the system, the test, and in large enough numbers that it has an impact, right? Because if you make one of them and that's being passed around the world, it's not going to help very much, right? So manufacturing is required. The other is that you probably have some type of regulatory approval that is needed. So in the United States, of course, that's FDA, but every country around the world will have some type of regulatory requirements, and many of them are quite high, as they should be. So we need to think about what that regulatory path is, what sort of studies need to be done. Outside of that, again, engaging stakeholders. Will people trust the technology? Will they adopt the technology? Hopefully that has come from early engagement, not something you do after the fact. Who's going to pay for it? I mean, obviously here we're talking about we're talking about serving low-income regions, but there's still a cost. Somebody has to pay, and in particular, a company has to manufacture, and so they will require payback for that investment. So understanding how that payment system works, who is incentivized to pay, who's able to make that decision to pay, is important. And then I've skipped the first part, but access to intellectual property. A lot of times in academic work, we might not think about that. Our group tends to think about it pretty early on, but often in academic work you don't. And so when you get to this stage where it needs to become a real product, you might find that there's a barrier to actually doing it because somebody's holding the patent that prevents you. Hopefully that's something that can be overcome, but it is something that you need to consider. Okay, so just a real quick examples here, or not examples, but the Harmony system that I described has actually been moved into commercial phase, and so this is the system, the test kit on the right, and then the detector, reusable detector on the left. It's taken a little bit different form. It's that black device that you saw earlier is now this green device. There's no cell phone. It just has a very simple screen. And the test kits, again, have everything that they need. Actually, it's almost the same as what you saw in the previous video. For any type of hardware, you have to figure out how you're going to manufacture it and do that in a reproducible way and a way that keeps costs down and can be done at the appropriate volume in terms of numbers of devices that you need. There's a whole lot of engineering work that has to go in at this stage. Simple things you might not think about, packaging of the test kits and any instrumentation, manuals that have to be written, customer support services that need to be provided, how you're going to get the tests out, to the people who need it, and in particular, as you get further out of the larger population centers, this type of distribution becomes more difficult. So having some plan or partnership in place, especially local partnership, would be very important for this. And then the manufacturing side, I want to emphasize this because people probably just don't have much experience. In fact, I didn't have experience with this before, but manufacturing it's a lot to undertake, and so it takes a lot of money and support and time and expertise to make manufacturing happen. And it has to be done in a way that maintains high quality of the product, consistency of the product. And then there's, for regulatory purposes, a lot of oversight that is needed to make sure that you're consistently doing the same thing and not deviating from it and creating a faulty product. There's been, in other contexts, like for used equipment that has been donated for global health purposes, some backlash because that equipment might be damaged. There may not be support to ensure that it keeps working, and so donated equipment a lot of times ends up not being used. And so you can imagine if you start to make a product and your quality starts to slip, and now you're providing an inferior product to your global health customers, that's obviously just not a good situation for health or for customer relationships. So this is a machine that its only job is to grab these folded boxes, stick them down on a conveyor belt here, unfold them, and glue the edges. And to actually get the test kit contents inside, somebody's got to go stuff their hands right there. Stuff the contents inside as these things flow by, which seems like a very dangerous thing to do to me. But this is a huge machine. That's all it does. And so there are lots of other machines that are needed to develop a manufacturing plant for this is for the test kits, but then also for the detectors. But it's also fun, I think. It's really interesting to see these things come together. Okay, so to kind of wrap things up here, just kind of looking forward, mentioned that communicable diseases are infectious diseases, and we heard this earlier, have been remained a problem in low-income countries. But that's changing, fortunately. There is progress being made on reducing the rates of communicable diseases. And unfortunately, though, it's being offset by increases in lifestyle diseases. So the types of diseases that come from poor eating habits, from sedentary lifestyle. So the things that we associate with our home here. I shouldn't say our home because I know people from everywhere, but from those are the problems that I grew up recognizing. So the heart disease and stroke that are often due to lifestyle choices, cancer for like lung cancer, for example. So unfortunately, there is a shift in that direction, but in terms of how we change our thinking, over your lifetime, we may be not talking so much about infectious diseases in terms of the magnitude of health impact, and might be, so there may be opportunities here to try to increase diagnosis and interventions for these chronic diseases. So thinking for developing technology here, simplifying existing technologies. If there's already something that's out there, how can we just do the engineering to make it simpler? Or taking a step back and asking really, for the particular settings we're talking about, is there really an innovative, different approach, that we should be taking for a health solution, right? Not just making a technology simpler, but really a health solution that really addresses the problem in a holistic way. Again, engaging local stakeholders is critical for understanding the needs up front, and ideally having, again, Dr. Farquhar talked about this a little bit, engaging people in a meaningful way locally in terms of technology development, and capacity building locally, manufacturing locally, having the business be centered in the places where it's going to be used. And then sort of broadly, I think going along with diagnostics is data science. So tracking disease, we heard a little bit about this just a bit ago, for both individual health and for overall population health, and then using that data to have specific interventions that can improve health overall. Okay, so I think with that, I'll wrap up. This is, I think I went through all of these. I think they're self-explanatory at this point. So I'd be happy, I think we got a minute for questions.
I think we'll have time for one question, Luis. So from the audience.
Okay, so one question from the audience is, how about non-infectious diseases like cardiovascular disease or diabetes or cancer? Like, how can these techniques you discussed in this presentation be used?
Yes, so absolutely they can. The question is about what is the biomarker? What is the thing that you need to detect? So for an infectious disease, obviously it's just something that shouldn't be in your body. It's a foreign organism. Cancer and diabetes, chronic diseases are a little harder because it's not necessarily, like cancer is mostly self. It's just a little bit changed. And so how do you tell the difference between the healthy cells and the ones that are a little bit changed? It can be done, but it's a little harder. For diabetes, other things that are involving metabolites that are normal metabolites, but their levels are off, may need like a quantitative measurement to as opposed to just is it there or is it not there. So it can be technically harder, but a lot of the same technical approaches that I talked about here could be applied there.
Thank you, Dr. Lutz. I think that's all the time we have for questions today, but thank you so much for joining. It's been an amazing learning experience, and I'm sure many of us will be inspired to pursue bioengineering as a career. So thank you for inspiring all of us today.
Great, thank you all so much. And you've inspired me.
Thank you.
Put this together. Yes, it's been amazing, and just so happy to see so much interest from young folks. Keep it up.
Thank you.